Hello, welcome to the YouTube channels for the some of the important lectures of biotechnology. We are very much aware that the biotechnology is the emerging branch, and this branch has been evolved for the welfare of the environment, society, organisms, and humans. And this is only possible due to one of the very important features that we can manipulate the DNA, we can manipulate the proteins. So, genetic engineering and the protein engineering, these two are the sections within the biotechnology by which we are getting the required DNA of our interest or proteins. So, how this DNA and the proteins we can get in the genetic engineering? Definitely, these processes they are regulated by the involvement of some of the enzymes. So in this lectures, I was I will be dealing with the enzymes for the DNA manipulation. So we are very much aware that the genetic engineering is the process of recombinant DNA technology, which deals with the cloning process. And for this cloning, we need one DNA from the A organisms, then they are inserted into the some of the vectors, and after that they are inserted into the some of the host cells like bacteria, and when this bacteria they multiply, then they make the two copies, then further they can multiply so that we can get the four copies like that. Not only that, the DNA belongs to any organism cell. They can be also allowed for the transcriptions and also for the expressions or the translation that the proteins. And for this we are using the cloning vector and the expression vector. So what are the different enzymes that are very much used in the biotechnology? In general, biotechnology is a set of techniques to manipulate the DNA or the protein as per our requirement, and it is only possible due to advancement in some of the technique like isolation of the DNA, getting a piece of DNA of required length because we have got the genomes, and from where how we can get the some of the fragments of this DNA, then insertion it in the heterologous organisms. Suppose that this gene it belongs to the mouse, and where we can replicate, where we can get that these two copies, so definitely we will insert them in the plasmid of the E. coli, and we can have the copy of the mouse gene. So this is the heterologous organism, and it is used for the cloning, that is making many copies, and the expressions, getting the protein product of the that DNA fragments. So what are the enzymes used in biotechnology? And this credit, it goes to the some of the important contributors in the field of biotechnology, and the very first name is the Paul Berg. So we can pay the homage and tribute to the father of genetic engineering, Paul Berg, who in 1972 succeeded in inserting DNA from a bacterium into the SV40 virus DNA. It means that the bacterial DNA has been inserted into the fast DNA. Later on, in 1973, biochemist Herbert Boyer and the Stanley Cohen that developed the genetic engineering by inserting DNA from one bacteria into the another cell, or that cell belongs to the foreign cells, not the same cells. And later on, the Stanley Cohen and the Herbert Boyer, they transplant genes from one living organism to the another, and this has led to the production of the human growth hormones and the hepatitis V vaccines. So we can say that the biotechnology is a boon for the society, and you know that in this corona pandemic, we are in the need of the vaccines, COVID vaccines, Covishield vaccines. Like so, how this is possible? So this is only possible due to the emergence of the biotechnology. We are using so many types of the biotechnological processes in order to get our desired requirement. So let us see that what are the different enzymes. So these enzymes are the catalytic biomolecules to initiate the any chemical reactions, whether it may be the metabolic reactions, that is anabolic, or it may be the catabolic synthetic reactions, or it is the breaking reactions. So RDT also involves the some of the enzymes, and these enzymes are template dependent DNA polymerase enzymes, then nuclease enzymes, where we can study the restriction endonucleotide, that's the main enzymes, and the N modification enzymes, then the ligase enzymes. So one by one we will try to list, but here I will only confine to the DNA polymerase, that is the template dependent DNA polymerase enzymes. So within this categories, there are a lot of the enzymes that are used in the our biotechnological processes. One is the DNA polymerase 1. It is the DNA polymerase 1, and the second one is the fragment. Third one is the T4 DNA polymerase, that is it has been isolated from the T4 phase, and the T7 DNA polymerase. So we know that this T phase, okay, tadpole like phase, that exists in the some of the odd T phase, and the even T phase. So odd T fast had been given the T1, T3, and even fast like that T4, T2, T6 like that. So T7 is the odd T fast, and T4 is the even T fast. And besides that, there is also the possibility that the cloning process or the synthesis of the DNA or the replication of DNA, that can be performed outside the cell, outside the cells replication, and also inside the cells. So in order to perform the replication outside the cells, we are using the thermocycler, or in better way we know that the PCR machines, that the polymerase chain reactions machines. So here we are using the thermostable DNA polymerase, because inside the cell, we can have the ability to just this double stranded DNA, they can be separated progressively, and simultaneously the replication goes on. Okay. But in the thermocycler, we need to increase the temperatures. And we know that when the temperature is increased, then the possibility that the polymerase, normal polymerase, they cannot sustain the high temperature, and they will just get denatured. So to avoid this denaturation of the protein, the thermostable DNA polymerase are very much used in the PCR, and that is known as Taq polymerase. And after that, but another polymerase activity has been performed by the terminal deoxynucleotide transferase. So let us see one by one how these enzymes work and what are the different properties. So let us that DNA polymerase. The DNA polymerase is the one of the important enzymes which can just have the either the RNA template or the DNA template. When the RNA template will be there, then it is called the RNA dependent DNA polymerase, and when it is the DNA, then it will be called as the DNA dependent DNA polymerase. Okay, so depending upon the template, they are accordingly named. So in eukaryotes, the DNA polymerase 1, this is the enzymes which are very much used in the biotechnology, and all these enzymes, for example, the DNA polymerase, these are the main enzymes which are very dominant in the in vivo condition. It is dominant in vivo condition. But when we want to use the enzymes DNA polymerase in the um, cloning purpose, then we will select the DNA polymerase 1. Okay, and these are the 3D structures where you can see the helix, beta pleated, and all other structures like that. So this DNA polymerase 1, as the name itself dictates that it is the DNA synthesizing enzyme. And for this DNA synthesizing enzyme, we always need the existing strand. So without existing template, the existing strand, the polymerizations cannot take place. So for the gene manipulations, one of the DNA polymerase one enzyme which is used is called the Kornberg enzymes, because it was named after the Arthur Kornberg, who are the discoverer of this very enzymes. And for this, they got the Nobel Prize in 1958. So this enzyme has got the both end exonuclease. It means that if it is the DNA and if it is the 5 to 3 strand, suppose that this has got the A, this has got the T. So if this has ability both end exonuclease, so it can remove it can remove the this A, and it can get the C and G and T like that. So this is the up 5 prime exonuclease. And suppose here it cuts, then it will be the CG, and then this is the T 3 prime. So that is called as the 3 dash or 3 prime exonuclease activity. So it pauses the both activity, that is the exonuclease from the 5 prime end and exonuclease from the 3 prime end. And beside that, it has also got the 5 dash to 3 dash polymerase activity. It is a hand-like structures. So Arthur Kornberg, when it discovered this very enzymes, so what are the some of the important facts? So this is encoded by differently the one gene that is called the polyagenes, the polyagenes, and this is a monomeric. This is this is a monomeric polypeptide that is consisting of the 928 amino acids, or inside a cell it has got the 400 molecules per cells, as compared to your pol 3, which is about 10 to the power 3, or like that 10 to This is, of course, it is wrong. It should be like 10 to 30. 10 to, sorry, 10 to 20. Okay. So it pauses the number of amino acids are 928, and the molecules per cell is 400, and the molecular weight is 103,000 इकाई दहाई सैकड़ा हजार दस हजार लाख. Okay, 1,03,000 Daltons, and this is the 10 to the power minus 6 base pair corrections. So it can, after one wrong base pair can be corrected after 10 to the power 6 base pair additions. So in order to know the what are the different properties of this Kornberg enzymes or the DNA polymerase 1, the protein fragments. These are the protein fragments. They are digested by the one enzymes that is called the subtilins. Okay, subtilin. When subtilins digest, then the uh, sensitive site is the serine site. That at the serine site, it breaks the peptide bond. Okay, when the peptide bond is broken, then what happens? It will leads into the small fragment and the large fragment. So this small fragment, it possesses the 5 prime to 3 prime exonuclease activity, while the large fragment, it has got the both polymerase as well as 3 prime to 5 prime exonuclease activity, and this activity is called as the proofreading activity. So proofreading activity that lies in the large fragment. The replication rate is 40 nucleotides per second. Okay, this is just a pictorial representation of the DNA fragment, where the DNA polymerase holoenzymes, which is a single polypeptide of 948 amino acids. This is called the polymerase. Green one is the polymerase domain. Blue one is the 5 prime to 3 prime exonuclease domain. Okay. So yellow and this one, this is the large one and this is the small one. So this is about 64 amino acids. Okay, 64, and this is about like 300 amino acids. So basically, this is very important. This is, and that's why this is called as the clinofragment. Clinofragment. The clinofragment is the large one, large fragment of 604 amino acids. This is again the pictorial representations of the clinofragment, where you can see this is the synthesis. Therefore, this is 5 dash to 3 dash polymerase activity. This is proofreading. This is the 3 prime to 5 prime exonuclease activity, and there is the primer removal. Primer removal. We know that a DNA is double helical. When it replicates, then it forms the single standard template, single strand template. Suppose that if it is the 3 prime and it is 5 prime, it is the 3 prime and this is the 5 prime. So initially with one primer, you can say the one primer it will synthesize, and after that the new strand of DNA is synthesized by the complementary base pairing. And in this strand, the primer, primer, primer. Okay, and after that the strands, and this is called as the Okazaki. This is called as Okazaki fragment. So this is what happened, 5 to 3. Okay, 5 to 3. Then what happens after the synthesis of this one? Then this is removed. This primer is removed. So this primer is 5 dash to 3 dash. So this is called the 5 dash to 3 dash exonuclease activity. Exonuclease activity. So here this is proofreading, and after that the lower one, this is 5 prime to 3 prime exonuclease. Okay, exonuclease activity. Sometimes, let's see. Okay, so what are the application of the clinofragments? So here's some of the applications I have enumerated. One is the synthesis of double stranded DNA from the single stranded template. Double stranded DNA from the single stranded templates. For examples, you are very much aware about the cDNA. Okay, it is the cDNA. Now, in order to insert this DNA into the vector, that this cDNA must be in the double stranded form. This is the double stranded cDNA. Okay, so how these are synthesized? So let us test this is the mRNA. Okay, this is the mRNA, and it will act as a template. Okay, and after that, the this strand is synthesized. Okay, so this is the RNA, and this is the DNA. So okay, this is the DNA. So this is possible due to the involvement of the one enzyme that is called as the uh, reverse transcriptase. Okay, reverse transcriptase. So this is the enzyme which involves the RNA template and synthesize the DNA strand. But this is heteroduplex. This is heteroduplex means one is the RNA strand, another one is the DNA strand. So after that, this RNA is degraded. This RNA is this RNA is degraded. This RNA is degraded, and this RNA, this RNA, sorry, this RNA. Okay, let me see. This RNA. Okay, so this is the you can say this is the degraded degraded RNA, and this is the DNA. Okay. So after that, this will act as now the template, and another new strand will be synthesized from 5 prime to 3 prime. Okay, 5 prime to 3 prime. Here it was 5 to 3, then this template was 3 to 5, again this 5 to 3. So this is possible by means of the clinofragment. Clinofragment. It means that the template was the single standard cDNA, and the double standard cDNA was formed due to the fragment, which was the large fragment of the DNA polymerase 1. Okay. Now the uh, second one is filled in the recessed 3 prime ends of the DNA fragments. So how we can explain this one? Again, we can explain that suppose that two strands are here. Okay, it is the A, C, T, A, G, T. Okay, and this is 5 prime to 3 prime. And the another strands are here. double stranded. This is again the T, G, A, T, C, T, and this is A, T, C, A. So this is called as the 3' recessed segment or the fragments of DNA, and this is called as the overhang. So how to make this one? So definitely this has to be filled. This has to be filled. So how this can be filled? This will be filled by the involvement of the clinofragment. Clinofragment. So this is called as the 3' excised 3 prime exist 3 prime filling in the resist 3 prime ends in the DNA fragment. Digestion of the protruding 3 prime overhangs. Okay, so this is the 5 prime to 3 prime. Now we can see that how it will possible. So let us see another fragments. The 5 prime to 3 prime. Okay, and this is the again the 3 prime to 5 prime. Okay, now so to make it this one, either we have to this one we can do, but it cannot be possible because if it will then the synthesis will be 3 to 5 prime. So this cannot be possible. This cannot be possible. So how this the two strands will make the blunt end? So definitely we have to just cleave or excise this one. We have to excise this one. So this is called the 3' overhangs excisions. Then when it will be excised, then we will have the this DNA. Okay. So in order to make the blunt end, this is called the staggered end. This is the staggered end. So we can staggered end DNA can be made blunt ended by the involvement of the clinofragment. So here what will happen? This is the 3' to 5', what is called 3' to 5' exonuclease activity, and that lies in the this is one in the large fragment. So all the time you remember that it has got the polymerase activity from 5 to 3. It has got the exonuclease activity, then 5' exonuclease, and the 3' exonuclease activity. Okay, so this one and this one it is in the large, and this one is in the small fragment. So third one, the preparation of radioactive probes by end leveling of the DNA. Okay, end leveling of the DNA. So if you want to just level the DNA, suppose that this is the N, this is the A or a T, you can level this one also, or you can this one. Normally, this is the this nucleotide. It has got the nitrogen 14. Okay, but if you want to have the nitrogen 15, then you will have to remove it, and then you will have to add it. You will have to remove it, you will have to add it. So that is only possible due to involvement of the clinofragment. Then random primer leveling of the DNA. Random primer. The primer can be also labeled by this one, and I will talk about this in vitro mutagenesis later on. So, and even the fragments are also used in the dideoxy chain termination method by means of the that is called the Sanger's method. Sanger's method, and of course the DNA amplifications or the cloning, where the DNA polymerase 1 is used to amplify the DNA. So this is of course the filling of the 3' disease end by the polymerase. So DNA polymerase, what is the source? So this, of course, the DNA polymerase one source is the E. coli. Okay, and of course it has got these three activity, and addition of in order to perform the polymerase activity, the end must have the 3' OH, that is 5 dash to 3 dash, it should be at the OH, then only the new nucleotide can be added. Filling the gaps in the double stranded DNA. Suppose that after the removal of the Okazaki primers, there will be the gap. So this gap can be filled by the this DNA polymerase 1. Of course, the synthesis of cDNA, separation or preparation of radioactive probes by N-labeling, and the DNA labeling by the Nick translation. Okay, Nick translation. So that I will tell you later on what is the Nick translation. Translation means the protein synthesis, but Nick translation is another one where the this is the strand, and after the cut is there, then what will happen? There is the degradations, and after the degradations, after the degradations, the new nucleotide is added according to the need. Okay, according to the need, or you can say the radio level. Suppose that initially it was having the A, C, T like that. So and this is the T, G, A. Okay, and ACT has been removed. So we have to add the labeling one. Then we will what will happen? This is the A leveled one, then C leveled one, then T leveled one. So unlabeled DNA strands can be made the labeled DNA by using the Nick translation. So Nick translation is the process of the DNA labeling. It is not the process of the translation. So after that, there is the another enzyme that is called the T4 DNA polymerase. It was it is isolated from the your what is called bacteriophage. It was isolated from the bacteriophage, and the function is again requires primed single stranded template. It means that if template is single stranded, then it can synthesize the new strand. It has also got the 3' to exonuclease activity, 5' to polymerase, similar to fragments. Catalyze the template dactyl synthesis from free 3' OH and bound to primer. Again, it is suppose that it is the primer, then it is 5 to 3, then it can add nucleotide here, and the processivity is the 4 nucleotide per second. So application is same, filling the in resist 3 DNA fragments, end labeling of 3' ends, end labeling of the 5' end, or also labeled DNA for use as a hybridization probe, labeling of the DNA, then making the blunt end DNA, and in vitro mutagens using synthetic polypeptide. So whatever the process functions that I dealt earlier, all these functions are performed by the this one. Another one is the T7 DNA polymerase. So this has been synthesized from the E. coli infected with the T7 bacteriophage. T7, T7 one when any bacteria, when this any bacteria, okay, it has been what you call transduced by the fast T7. Okay, so what will happen? It can either perform the lytic pathway or it can perform the lysogenic pathway. These two pathway. Okay, so either the this bacteria, when the DNA from the this T T7 that enters, then what happens? Either they will integrate this DNA will integrate here, or they will be okay, or they will replicate independently. Okay, so for these replications, they synthesize the polymerase. So that is called the T7 polymerase. Okay, the source is different. So you have to remember the source. That is the T7 DNA polymerase has been synthesized from the E. coli infected with the T7 bacteriophage. Again, it has got the 3' exonuclease activity, 5' 3' polymerase activity, then high processivity than other thermolabile bacterial DNA polymerase. Okay. Again, the end labeling, extension of the primer, DNA sequencing, that these all are the common functions. So now, another enzyme is the reverse transcriptase. Reverse transcriptase, I told you that this is the enzymes which are used for the cDNA synthesis. So this is called as RNA reactor DNA polymerase. Of course, another name is the Temin enzymes, and the process of reverse transcription is called the Teminisms after the discovery by the Howard Temin. Yeah. and Baltimore. So they both has got the credit and received the Nobel Prize in the year 1975. Another enzyme is the Taq DNA polymerase. So I told you that in the PCR, the we have to synthesize the DNA at high temperatures. Okay, we are synthesizing the DNA at high temperature. So why the DNA is synthesized in the high temperature? Because in order to synthesize the DNA, the initial double stranded DNA that has to be made the single stranded, that has to be single stranded, because they will also act as template, they will also act as template. So for this we have to denature the DNA, and the denaturation can be done either by the heat, or by the temperature, or by the some of the chemicals, for example ammonium hydroxide. It is also a denaturing agent. So this was discovered by the Chin et al. In 1976, and it was isolated from the Thermophilus bacteria that was named as the Thermophilus aquaticus, and the name that is the TAC, the initial TAQ. Okay, TAC polymerase. And this enzyme is stable at 70 to 90 degrees Celsius. When we isolate the DNA polymerase from the E. coli, so that is stable at the 37 degrees centigrade, or lesser than that. But high temperature, this enzyme will be degraded. So from the special or the specific bacteria, it is isolated. And of course, it has got the 5' to 3' polymerase activity, 5' to 3' exonuclease activity, but no 3' exonuclease activity. Okay, so you have to remember X3' to exonuclease activity has got the proofreading. So how the it differs? So DNA polymerase keleno fragment, and the what you call uh, fragment, and the this one Taq polymerase. This has got the 5' to 3' polymerase. It is also 5' to 3' polymerase. Second, it has got the 5' to 3' exonuclease. No, but it has got the 3' to 5' nucleus, but here 5' to 3' exonuclease activity is there. Okay, so 3' exonuclease nucleus activity is present in the fragment, but it is absent in the this fragment. So there are many type of the thermostable DNA polymerase enzymes are used. Basically, we are very accustomed to know about the Taq polymerase, which is about 94 kilodalton, and it was isolated from the Thermophilus aquaticus, and the relative efficiency is 88. Of course, the efficiency is very high, but if you see the error rate, then 10 to the power 2 into 10 to the power minus 4. So after every 10,000, the two base will be added wrong. Two base will be added wrong. So instead of this one, we have to go for the another one. So that though so many versions of the this thermophilus enzymes are there. Some of the that's Taq polymerase, TIL polymerase, PFE polymerase, TTGS polymerase. So these all are the polymerase. So another is that it has got the 3' to 5' exonuclease activity. Sorry, 5' to 3' iso. So once it will synthesize, then it can also degrade this one, and 3' to 5' exonuclease activity is not there. So proofreading is only possible. Proofreading cannot be possible in this one. Okay, no proofreading, no proofreading. Remember, the Taq polymerase, they can of course synthesize with more efficiency, but it doesn't have the 3' to 5' exonuclease activity, which is essentials to synthesize the correct DNA. So how this correct DNA will be synthesized? This correct DNA will be synthesized when the enzyme will possess the 3' to 5' exonuclease. But this enzyme has got the 5' to 3' exonuclease. So what will happen if the DNA is synthesized? Suppose that DNA is synthesized, this is the 3', this is the 5 template, and DNA is synthesizing. Okay, 5' to 3'. So what will happen if the A and the C is there? This is a long base pair. So unless and until this is degraded. How can the this correct nucleotide can be added? So in order to just remove this drawbacks, the another enzymes that is the PFU and the tilpol, they are used in order to correct the DNA. But now very much is the PFU is also used. So you must know that why the Taq polymerase, which one PFU and the other are better. These two are better than the this one. So because they don't have the Taq polymerase. Taq polymerase has no 3' to 5' exonuclease activity, which is the very essential for the proofreading. That is the removal of wrong base pairs. So here are some of the details that RTTH, it is the thermus thermophilus. This is the bacteria. It has been isolated from this one, and the enzyme has the reverse transcriptase activity. Reverse transcriptase activity in addition to 5' to 3' polymerase activity. So if the suppose that it is the 5' to 3' messenger RNA is there, messenger RNA there, then it will synthesize the RNA. It will synthesize the RNA from 5' to 3'. Okay, by this one polymerase. Okay, so basically, what will happen? It has got the two types of the activity. One activity is that this RTTH is I got the two types of the activity. It can act on the both RNA template as well as on the DNA template. Okay, so it can make the copy RNA DNA, and it will make the copy DNA DNA. This is the RNA, the DNA, and this is the DNA, and this is the DNA. So, uh, this is the had got the reverse transcriptase in addition to 5' 3 polymerase activity, and double standard 5 to 3' exonuclease activity is also here. Okay, and everything is possible in the presence of the manganese ions. Okay, so this has got the RTT has got the three activities. One this is the polymerase, and this is the exonuclease. The exonuclease activity is 5' to 3' exonuclease activity. Okay. Now, second one is the PFU. This has got the high fidelity thermostable enzymes approximately of 90 kilodalton isolated from the Pyrococcus furiosus. This is the source. Okay. And the enzyme replicates the DNA at 75 degrees centigrade. And what I told you about this one, because it has got the 3' to 5' exonuclease activity from the proofreading. Okay, this is the so these two, then tin Paul, it is a recombinant E. coli strain से निकाला गया है इसको, ठीक है, it is the purified from the recombinant. So this is the recombinant thermophilus enzymes. So when we will talk about the key, name the recombinant thermophilus enzymes, name the recombinant आपका thermostable DNA polymerase enzymes, then it will be the till Paul. So this is called as the vent DNA polymerase, and it has also got the high fidelity. Fidelity means the correcting DNA, synthesis of the correct DNA. Okay, so it is 5 to 15 folds higher than the observed data tag DNA polymerase. ठीक है, टैग पॉलीमरेज से इसका काफी ज्यादा 5 से 15 गुना आपको ज्यादा करेक्ट करने की आपको इसमें एबिलिटी है. एंड दिस हाई फिडेलिटी डिराइव्स इन पार्ट फ्रॉम द इंटीग्रल 3' टू 5' प्रूफ रीडिंग एक्सपेरिमेंट. तो इस रिकम्बिनेंट डीएनए में क्या किया गया कि एक 5' टू 3' पॉलीमरेज एक्टिविटी के साथ में इसको रिकम्बिनेंट बनाया गया कि इसमें 3' टू 5' एक्सोन्यूक्लियस वाला डीएनए प्रोटीन पार्ट भी इसमें ऐड कर दिया गया. एंड दैट वाई इट हैज greater than 90% of the polymerase activity in within the 1 hour or 95 degrees centigrade पे आपको किया जाता है, एंड इट इज ऑल्सो यूज फॉर द प्राइमर एक्सटेंशन सर। तो प्राइमर को एक्सटेंड करने के लिए, यानी कि टू मेक द कंप्लीट स्टैंडर्ड डीएनए, दिस इज यूज्ड। सो ऑल दीज एंजाइम्स आर द डीएनए पॉलीमरेज वन। Okay, DNA polymerase enzymes. So lot of this very enzymes are there. So at this we can say thanks, and so if any questions are there, so please share the your queries so that we can be able to answer your questions, and okay. So thank you very much. Uh,